Introduction to DNA. DNA, or deoxyribonucleic acid, is the hereditary material found in almost all living organisms, from bacteria to humans. It carries the genetic instructions essential for the development, functioning, growth, and reproduction of all known life forms and many viruses. Structure of DNA. Double helix model. DNA is structured as a double helix, famously described by James Watson and Francis Crick in 1953. The double helix resembles a twisted ladder. The sides are formed by alternating sugar deoxyribose and phosphate groups, and the rungs are pairs of nitrogenous bases. Nucleotides. The basic unit of DNA is a nucleotide, each consisting of a phosphate group, a deoxyribose sugar, one of 4 nitrogenous bases, adenine, thymine T, cytosine C, or guanine G, base pairing. The nitrogenous bases pair specifically. Adenine pairs with thymine T, guanine G pairs with cytosine C. These pairs are held together by hydrogen bonds, ensuring the stability and replication accuracy of the DNA molecule. Antiparallel strands. DNA strands run in opposite directions antiparallel, which is crucial for replication and function. Types of DNA forms. bDNA, the most common right-handed helix under physiological conditions. A DNA, right-handed, forms under dehydrated conditions. zDNA, left-handed, found in certain gene regulatory regions. DNA in the cell. Most DNA is located in the nucleus nuclear DNA, but a small amount is found in mitochondria, mitochondrial DNA. In plants, in plastids. Human DNA is organized into 23 pairs of chromosomes within the nucleus. Each chromosome is a single, long DNA molecule. Function of DNA. Genetic information storage. DNA stores instructions for making proteins, which perform most life functions and form cellular structures. Segments of DNA that code for specific proteins are called genes. Replication. DNA can replicate itself, ensuring genetic information is passed accurately during cell division. Each strand of the double helix serves as a template for creating a new complementary strand, a process known as semi-conservative replication. Protein synthesis. DNA sequences are transcribed into RNA, which is then translated into proteins. This flow of information is known as the central dogma of molecular biology. Genetic variation and mutation. Changes in the DNA sequence mutations can lead to genetic diversity, evolution, or sometimes diseases. RNA ribonucleic acid is a vital nucleic acid present in all living cells, playing a central role in protein synthesis and genetic information transfer. Here is a detailed overview of RNA. Structure of RNA. RNA is typically a single-stranded molecule composed of ribonucleotides, each consisting of 3 components: a ribose sugar, a phosphate group, and a nitrogenous base. The ribose sugar in RNA has a hydroxyl group, while attached to its defeat carbon, unlike DNA's deoxyribose sugar, which lacks this group. This makes RNA chemically more reactive and less stable than DNA. RNA contains 4 nitrogenous bases: adenine, guanine G, cytosine C, and uracil U. Uracil replaces thymine found in DNA and pairs with adenine via 2 hydrogen bonds. Although single-stranded, RNA can fold onto itself through intramolecular base pairing, forming complex 3-dimensional structures such as hairpins, bulges, and helices, which are crucial for its function and stability. RNA molecules can be chemically modified, for example, methylation to stabilize their structure and enable specific functions. Types of RNA and their functions. RNA exists in several types, each with distinct roles in the cell. Messenger RNA mRNA carries genetic instructions transcribed from DNA in the nucleus to the ribosomes in the cytoplasm, where proteins are synthesized. mRNA is relatively short and single-stranded. Ribosomal RNA, rRNA, the most abundant RNA. It combines with proteins to form ribosomes, the cellular machinery for protein synthesis. rRNA constitutes about 50% of the ribosome structure. Transfer RNA. RNA has a complex folded structure and transports specific amino acids from the cytoplasm to the ribosome during protein synthesis, matching amino acids to the mRNA codons. Functions of RNA. RNA's primary function is to translate genetic information from DNA into proteins, which are essential for cellular structure and function. It acts as a messenger carrying instructions from DNA to ribosomes, facilitates the assembly of amino acids into proteins, and forms part of the ribosome structure itself. Some RNA molecules have catalytic activities, ribozymes, and can act as biological catalysts, a role traditionally attributed only to proteins. Chemical properties. The presence of the 2-hydroxyl group in ribose makes RNA more prone to hydrolysis, contributing to its shorter lifespan compared to DNA. This chemical ability is one reason DNA is the preferred long-term genetic material in most organisms. RNA's ability to fold into complex shapes and form a ribonucleotide complexes is essential for its diverse functions in the cell. Key difference between DNA and RNA. Strands and structure. DNA is double-stranded, forming a double helix that provides stability for long-term genetic storage. RNA is typically single-stranded, allowing it to fold into various shapes for different functions. Sugar difference. DNA contains deoxyribose sugar, which lacks an oxygen atom at the 2' carbon, making it more chemically stable. RNA contains ribose sugar, which has a hydroxyl group at the 2' carbon, making RNA more reactive and less stable. Bases. DNA uses thymine T as a base, whereas RNA replaces thymine with uracil U. Both pair adenine with their respective pyrimidine bases a T in DNA. A U in RNA. Function. DNA serves as the long-term storage of genetic information. RNA acts as the messenger and functional molecule that translates this information into proteins, including mRNA, tRNA, and rRNA roles. Location. DNA is primarily located in the nucleus and mitochondria, while RNA is found throughout the cell, including the nucleus, cytoplasm, and ribosomes where protein synthesis occurs. Stability. DNA's double helix and deoxyribose sugar confer greater stability, suitable for genetic storage, when a single strand and ribose sugar make it more chemically reactive and short-lived, fitting its role in transient genetic messaging. These differences underline the complementary roles of DNA and RNA in cellular biology and genetic information flow. Thank you for listening. Don't forget to subscribe my channel.